what you're building at Pauling.ai, and what made you take the leap from big tech into drug discovery?
One of them is, maybe serendipitously because, there's some substance in nature that seems to have some effect, and then scientists, take it, purify it, test it in different ways, and then they apply it as a medicine, right? So many times, antibiotics, for example, were found like that, right? Penicillin. I think there's a story about Alexander Fleming. Had discovered it in the lab, realized that it killed bacteria, and so on. Most drugs, maybe not most, but a big chunk of them were found that way. The other way that drugs have been discovered, especially recently, is using what's called rational drug design, in which we use what we know about molecular biology, about how different molecules interact inside of a cell and what causes a disease, and then we find other molecules that may work very well for or interact in a specific way with some of these other molecules that are inside of a cell. So to give you another example, maybe we understand a little bit about cancer biology, and then we know that cancer cells avoid the immune system. They do that using certain surface proteins in the cell. So we can go and say, okay, or the scientists, could say, if we could prevent those proteins in the surface of the cell from binding to something else, then the immune system would find that cancer cell and then would kill it. And that's kind of the origin story of the what's called today checkpoint inhibitors, so like PD, like PD1, PD-L1 inhibitors. So then somebody can go and say, well, let's find a monoclonal antibody, for example, that binds to that specific PD1 protein, and then the immune system would go and kill that cell. So there is a systematic process that you would follow from identifying a specific target to designing or finding a molecule that binds to that target. And that is, the initial phase of, what's called drug discovery. Now, there are, of course, many other things that are required, right? So once somebody has a molecule that could be used as a medicine, there's all sorts of things that need to happen, experiments on cells and animals or organoids or all sorts of different things. And once there is enough, I would say, certainty about the safety of that molecule, one could begin, clinical trials, phase 1, phase 2, phase 3, like there's, years of clinical trials that need to happen. But that very first stage is basically going from an idea about a potential therapy to a molecule that can be tested. Now, that process typically is very time intensive. And there are, it's done sometimes at universities, sometimes in companies, right? But it is one of the big, I would say, roadblocks to developing new medicines. It takes years, and it costs a substantial amount of money, and it requires a certain level of expertise. It is not something that a regular biologist or an MD or somebody with a knowledge of a disease could do. It requires a very, very deep understanding of molecular biology and different ways of designing molecules or finding them and so on. So what we have done at Pauling is we have created an AI agent. You can think of it as a ChatGPT, kind of tool that does many of those same tasks in early stage drug discovery. So you come to the tool and tell it, I'm interested in this specific bacteria, for example, and then we would identify a protein that is a potential target to kill that bacteria, and then find a small molecule, an antibiotic would typically be a small molecule, find a small molecule that binds to that target, and we do that in a fully automated way. So somebody that wants to go through that process could be a biologist, for example. They don't have to understand computational chemistry. They don't have to understand how to set up all these tools, how to run experiments, how to analyze those experiments, how to orchestrate everything together, how to, download and massage all the data, all the things that somebody would typically have to do, which is part of the reason why the process takes, many months or years. So what we have done is essentially automate that entire process and put it behind an interface that looks a little bit like ChatGPT, little text window and so on. But our tool, different from ChatGPT, can run a lot of computational chemistry tools, right? So a lot of, we don't need to get into the details, but like what people, typically use for this type of tasks, docking, molecular dynamics, admit, prediction models, and so on and so forth.
So basically what you're saying is, so in your case, you're actually not using AI to sort of replace scientists. You're just giving them that computing power and the pattern recognition that earlier would sort of take sort of years to do, and then they can do it faster now.
Yeah, it's basically giving a tool for scientists to do early stage drug discovery, but much faster and in a much more comprehensive way than they could before. So, if you think about it, the same way that you could go to if you're a writer, right, and you need to do research for an article, you can go to ChatGPT or you can go to Claude or any of these models and say, hey, I'm doing some research for this specific article. I want you to crawl all these links, gather all this information, structure it in a way, and then you can do the last analysis, for example, and then write it in your own voice, right? So it's the same way that, before scientists would have a team of computational chemists and biologists and people that had experience doing these things, or they would hire a company to do it for them. And, they would set up the project, with the constraints that they need. And then the company would come back and say, here's my list of top hits. So what we have done is basically replacing that, right? But ultimately, of course, there has to be a scientist that understands a little bit about what is the disease that we want to tackle, how we're going to do that, and kind of validate that the model is performing well. But, not a team of a dozen people, right, as before, right? But, hopefully, just a single person, can drive this entire process.
If you sort of have to break it down, so for most people listening, R&D is still kind of a black box, and everyone just kind of wonders every time, why does it take for a new medicine to sort of come out in 10 to 15 years, and why do we spend billions of dollars? And of course your mission does help mitigate those costs and timelines to a bit, but if we kind of break it down, where exactly does AI fit in the whole process?
Yeah, I mean, so I think if you look at the different stages that are involved here, right? So there's maybe a part of what's called in silico drug discovery, which is typically done with computer simulations that may take a year. Then there is maybe preclinical work or IND enabling work, which is typically done in cells, different types of biochemical assays, structural assays, animals, and so on and so forth. And then part of the reason why that second phase takes a while is because sometimes the biology takes a while, right? So if imagine, for example, that you want to find a cancer medicine, right? And you need to take an animal with a cancer. Typically, that tumor is grafted inside of that animal, so you need to do that, and then wait a little bit, make sure that animal is still is alive, right? Then you apply the medicine, and then see what happens to that tumor. And then you need to wait a certain amount of time to see if the tumor comes back or not, right? So that entire process may take a few months, right? It takes a few months instead of years because typically these experiments are done on mice, right? That may live, 2-3 years, right? So people would typically check that the mice recovers, for example, and then after a few months, the cancer doesn't come back. Now, quite often that experiment doesn't succeed at the first try, right? Part of the problem is that, people don't do all the simulations that they could do at the beginning, right? So they find out, a little bit later that the molecule is not as good. So what happens is somebody has to come back and then make some changes and then try a novel molecule. So every time that you do one of these iterations, it takes several months, right? So if you need to, go back 2 or 3 times, right? 3 times 3 months, plus a little bit of padding, that is basically 1 year. right? That you spend, doing experiments on animals, even after you have already a molecule.
3 months and then 3 mice as well.
Well, yeah, I mean, like, you would probably use a few dozen, for each experiment, plus controls, plus, maybe positive controls and all of that. So, like, fortunately, these things require a lot of animals as well.
Yeah.
But also people do things in different phases, right?